Hello everyone, welcome to the new session of Polyploidy. In this session, we are going to study about the Polyploidy. In the previous session, we have studied about Allopolyploidy, right? And in this session, we are going to study about the Aneuploidy and Autopolyploidy. Okay, so this is the second session for the Polyploidy. Okay, so let us start this session. Aneuploids are the polyploids that contain either an addition or subtraction of one or more specific chromosomes to the total number of chromosomes that usually make up the ploidy of a species. So here the chromosomes from the original number, that is the total number of chromosome, either a specific chromosome will be less or an addition of a chromosome will be there. Okay, so an addition or subtraction of one or more specific chromosome to the original number of chromosome, if in an organism is there, then that particular phenomena is called as aneuploids. That is called as aneuploids. So aneuploid results from the formation of univalent, okay, and multivalent. Suppose any organism that is having the 7 chromosome and it is having 7 pairs of chromosome like this. Okay, so this is the 7 pairs of chromosome, pair 1, 2, 3, 4, 5, 6 and 7. So if one of the chromosome is less, then this is not paired. This is not paired. So that is, it is called as univalent. And if it is paired and one more extra chromosome of this particular type, that is chromosome number 1 is there, then it will be multivalent. So let us see in more detail how the aneuploids are formed. For example, several studies have found that 30 to 40% of the progeny derived from autotetraploid maize are aneuploids. Okay, so from the autotetraploid of maize type, 30 to 40 progenies are aneuploids, okay, and with no mechanism of dividing univalents equally among the daughter cells during the anaphase 1. Some cells inherit more genetic material and other less. So if we see this previous case which we have discussed in the previous slide, at the time of gamete formation, again I am drawing that. So this is the 1, 2, 3, 4, 5, 6, 7. At the time of gamete formation, this particular gamete will have 6 chromosomes and this another one will have 7 chromosomes. Because here one chromosome has been lost. Okay, so now this type of gamete when unite with the normal gamete, okay, and this fertilization takes place, then what will happen? This particular chromosome will be less. This particular chromosome will be less, and again there will be unpaired number of chromosome. Okay, so in this form the aneuploids are formed. Similarly, the multivalent such as the homologous chromosomes may fail to separate during the meiosis, leading to the unequal migration of chromosome to the opposite poles. And again, how the multivalents are formed? Multivalents can form in this way. So at the time of meiosis, the chromosomes are arranged like this. Okay. So spindle fibers are attached to the poles. Okay. And what happened? These chromatids, this centromere splits and chromatids moves to the poles. Chromatids moves to the pole. But sometime this centromere fails to divide, and this whole chromosome, both the chromatids move toward one side. Okay, and they will have, this particular gamete will have one extra chromosome of a particular type, and this one will have no chromosome of that particular type, right? Suppose this is chromosome number 1, 2, 3. I'm just taking 3 chromosome here. So the second one, this is moving towards this particular pole. Okay, and because the centromere do not divide here, so this chromosome will move toward this side, or it can move to this side also. So the spindle fiber helps to move this whole chromosome towards this particular nucleus, and the nucleus will form here, and this whole chromosomes will be present in this particular nucleus. Okay, but in what happened to this side? This side, there will be no chromosome. There will be no chromosome number 2. There will be no chromosome number 2. Okay, so this is called non-disjunction of the chromosome. This particular phenomena is called as the non-disjunction. Okay, so this multivalent such as the homologous chromosomes may fail to separate. Here it fails to separate. This particular time it fails to separate during the meiosis, leading to the unequal migration of chromosome to opposite pole. So here it is unequal migration of chromosome, and this is called as non-disjunction of the chromosome. So these meiotic aberrance results in the plants with reduced vigor. So here some of the examples of the aneuploids. Okay, aneuploids are classified according to the number of chromosome gained or lost. So it is called monosomy when there is 2n plus minus 1. Any of the chromosome is present in a single form, not in a paired form. Okay, so this is 2n minus 1. Nullisomy, both the chromosomes are missing. One of the paired chromosome is missing. Okay, trisomy, so apart from this 2n, there is one extra chromosome. In the case of tetrasomy, 2 extra chromosome from the normal chromosome is present, and in the pentasomy, 3 extra chromosome is present along with the normal chromosome. So according to the number of chromosome missing or gain, the terms are defined: monosomy, nullisomy, trisomy, tetrasomy, and pentasomy. Now let us discuss about the autopolyploids. So autopolyploids are referred to as autoploids. They contain multiple copies of the basic set of chromosome in the same genome. Okay, so the autoploids occur in nature through the union of unreduced gametes at the time can be artificially induced. So suppose the basic set of chromosome number is 12. Okay, so we could have 36 chromosome in the one of the autopolyploid example. So 12 plus 12 plus 12, 3 complete pair of chromosomes are present here, and that forms the 36 chromosome. 36 pairs. Okay. Natural autoploids include tetraploid crops such as alfalfa, peanut, potato, and coffee, and triploid banana. So these are some of the examples of tetraploid, that is a type of autoploids. Okay, naturally they are present as a autopolyploid. Okay, so alfalfa, peanut, potato, and coffee, these are some of the examples of the autopolyploid that are formed naturally. Okay, they occur spontaneously through the process of chromosome doubling. So every time the chromosome gets double at the time of gamete formation. Okay, and what happened when the new offsprings are formed, they will have a complete set of chromosome increased. Okay, every time. So the chromosome doubling in the autoploids have very effect based on the species. They will have the varying effect based on the species. Spontaneous chromosome doubling in ornamental and the forage grasses has led to increased vigor. So some of the examples are there that undergo the spontaneous chromosome doubling, like some of the ornamental plants and the forage grasses, okay, that have increased in vigor, so they are favored by the environment. For instances, ornamentals such as tulip and hyacinth and the forage grasses such as the rye grasses have yielded superior varieties following the spontaneous chromosome doubling. So these are some of the ornamental plants examples that undergo the spontaneous chromosome doubling and leading to the phenomena of the autopolyploidy. Okay. Due to the observed advantage in nature, breeders have harnessed the process of chromosome doubling in vitro through introduced polyploidy to produce superior crops. Now, this particular thing, that is the chromosome doubling, okay, spontaneous chromosome doubling, which was occurring in naturally, now the plant breeders has adapted that chromosome doubling in vitro, means in the laboratory. Okay, an induced polyploid phenomena takes place and forming some superior crop varieties. For example, induced autotetraploids in the watermelon crop are used for the production of seedless triploid hybrid fruits. Okay, watermelon crop, and there is also the banana crop example. Very important. Okay, so these are some induced autotetraploids that has been developed in vitro. Such polyploids are induced through the treatment of diploids with mitotic inhibitors such as dinitroanyls and colchicine. So these are some of the chemicals that are used for the treatment of diploids with the mitotic inhibitors. Okay, they inhibit the mitotic spindles and lead to the formation of diploid gametes. To determine the ploidy status of induced polyploids, several approaches may be used. They include chloroplast count in the guard cells, morphological features such as leaf, flowers, or the pollen size, or flow cytometry. So these are some of the approaches that is used to determine the ploidy status in the polyploids. Okay, so let us discuss about the induced autopolyploids. So let us consider a plant that is having 2n is equal to 14. So at the time of gametogenesis, what happened? No spindle fiber formation at the anaphase 1 takes place. During the time of meiosis, and all homologous chromosome will migrate to the same cell. As a result, diploid gametes formation takes place. Okay. Now, after the fertilization with another diploid gamete gave rise to autotetraploid. Autotetraploid means 2n is equal to 28. Okay. Now, the zygote divide by the mitosis process, so descendant will be tetraploid. So all the cells will form now tetraploids. So the main thing here, that during the time of anaphase 1, no spindle formation takes place due to the treatment. Either it is a naturally occurring phenomena, or with the help of some chemicals like colchicine, the spindle fiber formation does not take place at the time of anaphase 1, okay, of meiosis. and homologous chromosome will migrate to one particular cell only, leading to the formation of diploid gametes. Now such a diploid gamete when fuses with another diploid gamete, then it will form a zygote that is having 28 chromosome, that is autotetraploid. Okay, so the zygote when divide, it will form all the cells that are having 28 chromosome number. So that is the example of formation, how the tetraploids are formed. So one of the example is the triploid banana. So let us see how it is formed. The ancestors of the cultivated banana are the diploid species, that is Musa acuminata and Musa balbisiana. Okay. So naturally occurring the intraspecific and interspecific hybridization led to the various combination of A and B genome. So this is representing the genome of Musa acuminata, capital A. This is the genome of Musa acuminata, and capital B is the total genome of Musa balbisiana. Okay, so what happened? Sometime we can have this or we can have this. Okay, this is the intraspecific hybridization. Okay, and sometime interspecific hybridization also. Let us see how it happened. So most edible banana are the triploids with genome capital A, that is the desert, and capital A, capital B, that is the plantains, and capital A, capital B, that is the cooking. So these are certain edibles banana, okay, in the form of triploids. Now multivalent formation means bananas are seedless. So this is one of the advantage of the multivalent formation of banana, that they are seedless. And seedlessness is a good for the consumer but problematic for developing new varieties. The only propagation method that will be asexual method. Okay. So the ancestors of the modern cultivated banana are both diploid species. Both have same floral structure. Okay, and the floral structure persists in the triploid banana, that is 2n is equal to 33. Flowers are functional in diploid, but triploids do not experience a successful fertilization. Now in the triploid form, the flowers are not functional. Right? The flowers are not functional. But in the case of diploid, they have functional flower. So if the flower will not functional, then there will be no fertilization. Right? So a possible model for the origin of triploid banana is Musa acuminata, that is the capital A spontaneously doubles, that is capital A and capital A form. Okay, so this is the spontaneous doubling of its genome. Okay, and here we are having capital A or capital B. Either it will be Musa acuminata or Musa balbisiana. Any species there could be. Okay, so From this, at the time of gamete formation, here the gametogenesis takes place, and these are the gametes. These are the gametes. So if it will be A and A, then the gamete will be capital A, and if there is capital A, capital A, capital A, capital A, then the gamete will be capital A type. And when there is a fertilization process, then what happened? This capital A will fuses with the capital A, or if it is Musa balbisiana, then it will be capital B. So here we can have capital A combination, or capital A capital B combination. Okay, so the triploid banana then would be the propagated asexually. So what will happen here? No fertilization in these type of induced autoploid. Why? Because flower is not functional, and that due to that they have to undergo asexual reproduction. Okay, 90% of the desert banana, that is the capital A is the world are genetically identical. These are the genetically identical, and that is variety of Cavendish. They undergo strictly for asexual propagation. They follow asexual propagation. And Cavendish is highly susceptible to the Siketoka disease, that is a fungal pathogen that is caused by the fungal pathogen. So this is all about the polyploidy. Okay, so we will meet with one more interesting session. Till then, thank you very much.